First speaker is Dr. Minamikawa Kazunori, Senior Researcher of the Crop, Livestock, and Environment Division of JIRCAS. Dr. Minamikawa is promoting the use of alternate wetting and drying technology for paddy rice fields. And today, he'll talk about climate-smart rice production by alternate wetting and drying.
Hello, everyone. My name is Kazunori Minamikawa. My major is soil science and agronomy. I'm studying the mitigation of greenhouse gas emissions from a paddy field for more than 20 years. Today's my topic is AWD, alternate wetting and drying, as a climate-smart technology for rice production. As a background for this study, rice cultivation is a major source of atmospheric methane. Meanwhile, rice cultivation is also affected by changing climate, such as high temperature, elevated CO2 concentrations, and extreme events. Therefore, we should cope with both climate change mitigation and adaptation through climate-smart technologies for rice cultivation. As you know, about 90% of global paddy areas are located in Asia. It means that global methane emission mostly comes from Asia. Actually, rice cultivation is the dominant source of agricultural methane emission in 10 countries of Asia. For example, in Cambodia, the contribution of paddy methane emission to the national total GHG emission is very high. In the Philippines, Thailand, and Vietnam, the contribution is also more than 10%. This schematic diagram briefly shows the mechanisms of paddy methane emission. Under strictly reductive soil conditions developed by continuous flooding, methane is produced from organic carbon by microbes. In contrast, during drainage, oxygen is supplied into the soil, thereby stopping methane emission, but instead increasing nitrous oxide emission. Therefore, appropriate drainage practices can mitigate paddy methane emission. This schematic diagram shows the seasonal changes in surface water level by continuous flooding and AWD. As far as I know, continuous flooding is implemented as a conventional practice in many Asian countries. In contrast, the dissemination of AWD is underway. AWD is a multiple drainage practice based on surface water level that has been developed by IRI, International Rice Research Institute, for saving irrigation water. When the water level naturally declines to a predetermined threshold, field is irrigated. This cycle is repeated, except for the heading stage. Many field studies have confirmed that AWD using -15 centimeter threshold achieves no yield reduction compared to continuous flooding. How are the quantitative effects of AWD on GHGs and rice yield? This table shows the results of recent meta-analysis papers. Red color indicates negative value, and blue indicates positive. As you can see, methane emission is decreased. Nitrous oxide emission is increased. And CO2 equivalent emission of methane and nitrous oxide is decreased. but the effect on rice yield is not consistent among studies. This table summarizes the results of the last table. The results for GHGs are consistent with our existing knowledge. But please note that nitrous oxide emission enhancement can be reduced if the field is flooded during nitrogen fertilizer top dressing. And for rice yield, someone may think AWD decreases rice yield. Others may think AWD has no effect. And I think AWD has a potential to increase rice yield. This is because at 3 locations in Vietnam, JIRCAS found that AWD can significantly increase rice yield relative to continuous flooding. If so, why? Under double or triple cropping systems in lowland, paddy soil is often under reductive condition. So the first hypothesis is that AWD may increase rice yield through ameliorating the reductive soil conditions. But as you may know, rice yield increase by AWD is not always observed. So the second hypothesis is that careful drainage practice considering rice growth stage may increase rice yield under other soil conditions. What is a careful drainage practice considering rice growth stage? MIDI, mid-season drainage followed by intermittent irrigation, is a common practice in Japan. This is rice stage-dependent water management practice aiming at sound growth and yield increase. And it has a long history. In the late tearing stage, mid-season drainage is implemented to reduce toxic substances in the soil and to remove ineffective tillers. After the mid-season drainage, intermittent irrigation is repeated on a weekly basis, except for the heading stage. This can enhance the resistance to rotting and can promote grain filling and carbon translocation. Now, JIRCAS is carrying out multi-year field experiment at 7 sites under diverse rice environments in Asia. Our objectives are to test the 2 hypotheses and to elucidate the mechanisms of rice yield increase by AWD and MIDI. I think that AWD is already a climate-smart technology through reducing methane emission and saving irrigation water. But if AWD can increase rice yield, I believe that AWD will be voluntarily disseminated to farmers. This is my final slide. In the near future, we expect to elucidate the determining factors and limiting factors in rice yield increase by AWD and MIDI. Establish quantitative relationships between drainage intensity, GHG emission, and rice yield. And finally, publish practical guidelines for rice yield increase by AWD and MIDI. Beyond this field study, JIRCAS will try to disseminate AWD and MIDI to Asia as a climate-smart technology for rice cultivation through 2 approaches. First is the voluntary approach, For example, through demonstrating rice yield increase as an incentive for farmers. Second is the institutional approach. If we can develop a new methodology of monitoring, reporting, and verification that can minimize the transaction cost, the dissemination of AWD and MIDI will be accelerated in the context of Paris Agreement Article 6. That's all. Thank you for your kind attention.
And the next speaker is Dr. Guntur Subbarao, Senior Researcher of the Crop, Livestock, and Environment Division of JIRCAS. Dr. Subbarao pioneered the development of biological nitrogen inhibition, or BNI technology, and was awarded the 2021 Cosarelli Prize by the National Academy of Sciences of the United States for his research on BNI-enhanced wheat. Today, he'll give a talk on BNI-enhanced wheat and maize, which maintain the yield with reduced nitrogen fertilizer application.
Thank you, Mr. Chairman. My name is Subbarao. I've been working as a senior researcher in JIRCAS from the last 20 years. So the topic I'm going to discuss here is the BNI technology and what it means for environment and agriculture, and specifically addressing, can the BNI-enabled wheats maintain grain yield with reduced nitrogen fertilizer applications? That is the topic, that is the case study we are going to show during this talk. So if you look at the nitrogen fertilizer applications, they have grown nearly 30-fold from the last 6 decades, since the beginning of Green Revolution, where the nitrogen fertilizer applications drove the Green Revolution, which is the main driving factors for the Green Revolution in transforming the agricultural productivity. And since then, in the early 1950s, in the beginning of Green Revolution, the global nitrogen fertilizer usage was about 5 million metric tons, now which has grown into 150 million metric tons, a 30-fold increase. So during this 6 decades of Green Revolution, the agricultural productivity has grown nearly 3-fold. But the nitrogen fertilizer application have grown nearly nitrogen fertilizer consumption has grown nearly 30-fold. So the problem, you can see that the nitrogen use efficiency in agricultural production systems is on continuous decline. And nearly 70% of the nitrogen fertilizers we are applying in agricultural systems are being leaked out from the farmlands, causing enormous pollution problems in the form of nitrate pollution of groundwaters and nitrous oxide emissions from agricultural systems. And the point is, the issue is why so much nitrogen fertilizers are leaking from our farmlands. When we apply nitrogen fertilizers, we are not only providing nutrition to the crops, but there is also a large group of soil bacteria living in the soils, which is using this nitrogen fertilizer as a source of energy and transforming into other harmful forms of nitrogen. And that is one of the reasons why the nitrogen use efficiency is becoming lower and lower with time. And you look at this, the soil, which is a living biological system, a group of bacteria called nitrifying bacteria that uses nitrogen as a source of energy and convert the nitrogen into nitrate and other harmful nitrogen forms, once the nitrogen, most of the nitrogen fertilizer what we apply is in urea or ammonium form, and the nitrogen, as long as it is in ammonia or urea form, it binds to the soil and stays in the soil. It doesn't leach out, it doesn't leak out. But this bacteria in the soil that oxidizes this ammonia into nitrate. And once it is converted into nitrate, this nitrate cannot bind to the soil and is also susceptible to the gaseous converting into the transforming into the gases. And that's the reason once the nitrogen is changed into nitrate, there's very little we can do. Maybe it's very difficult to keep the nitrogen in the soil. And that is one of the reasons why it leaks out of the farmlands. And nearly, as I said, nearly 70% of the nitrogen fertilizers we are applying to agricultural systems are being leaked out. The economic cost from this nitrogen fertilizer loss is about $70 billion US annually from the global nitrogen, from the global agricultural systems. The environmental cost is much higher from these nitrogen losses, which is estimated at about 70 to 320 billion euro from EU alone, which uses less than 10% of the global nitrogen fertilizer. So if you actually look at the environmental and ecological cost of this nitrogen pollution, which is much, much higher than the economic cost, what you see, the direct nitrogen from the direct nitrogen fertilizer loss. So if we keep the nitrogen in ammonium form, it doesn't cause any kind of harm to the environment. It doesn't leak out from the soils. But once it is changed into nitrate form, that is where most of the environmental problems and economic inefficiencies are coming into picture. So the question is, and once it is converted into nitrate, you can't keep the nitrogen in the farmlands. It leaks out and it ends up in the water bodies, causing this kind of nitrogen pollution. And also most of the nitrous oxide is generated from the farmlands, which is coming from the denitrification of nitrate. And most of the nitrous oxide, that is emissions coming from the farmlands, started from the beginning of the Green Revolution. And you can see that now the nitrous oxide emissions globally is increasing in a linear fashion. And nearly 20% of the nearly 20% increase in nitrous oxide levels in the atmosphere in the last 60 years happened, and it is going to go up even further, which is going to contribute to the climate change and the greenhouse gas, the global warming. So the challenge is how to keep soil nitrogen within farmlands. That is the biggest challenge before the agricultural scientist community now, because to increase the food production, to meet the food production needs by 2100, it is expected that the nitrogen fertilizer applications will have to be doubled if we have to follow the current nitrogen fertilizer technology. So, which is going to be a major disaster on the environment and the ecosystem. So, my talk is going to be how this new DNA technology, what we are developing, would work, and the effectiveness of BNIs, and how significant this kind of new technology in wheat production systems, and what is the current state of progress and the future direction. And the BNI function, BNI is a biological nitrification inhibition. What we are going to say is that what is the basis of this BNI is that the plant root systems naturally produce these nitrification inhibitors. It's a kind of antibacterial compounds coming from the root systems that control this nitrifying bacteria in the soil. So these antibacterial compounds can control the nitrifying bacteria in such a way that they will not allow them to function. And because of that, the nitrogen would remain in ammonium form, which is available for the crops to take it up. So once you control the flow of nitrogen from ammonium into nitrate, most of the nitrogen problems, what you see from the nitrate leaching or nitrous oxide generation, will be reduced or stopped. That is the concept of BNI function. And if we can improve, we can make the crops to produce this kind of antibiotics from the root systems in such a way that we can develop the next generation crops that can control this nitrifier activity in such a way that keep the nitrogen more in ammonium form and less in nitrate form. That is the BNA technology. And this can be genetically manipulated to produce the next generation crop varieties. So the plants merely produce spend nearly 30% of the carbon through the root exudation. So basically they produce two categories of nitrification inhibitors. Nitrification inhibitors is nothing but kind of antibacterial compounds that are specifically directed at this nitrifying bacteria in the soil. So by improving the production of these nitrification inhibitors or the antibiotic compounds, we can control the nitrifying bacteria in the soil so that the nitrogen leakage can be stopped. So our group in JIRCAS has isolated a variety of nitrification inhibitors that is coming from various crop species, starting from sorghum or Brachyara pastures, or recently our group has isolated a number of these nitrification inhibitors from the maize root systems, what you see over here. And also, I'm going to show some a case study where we have how we have improved the nitrification inhibitor production from the wheat root systems by transferring a chromosome segment that controls this inhibitor production from a wild wheat into the cultivated wheat. And when we look at the nitrification inhibitor production from the cultivated wheats, it's not sufficient to make a big difference in the field. But when we look at some of the wild species like the Limbus racemosus, it can produce large amounts of these nitrification inhibitors, these antibacterial compounds from the root systems. This is the focus of this was the focus of our work from the last 10 years, trying to transfer this kind of ability into the cultivated wheat. And the ability to produce these inhibitors is located in chromosome N and short arm of wild wheat. Our group has successfully transferred into the cultivated wheat using the chromosome engineering techniques. And this is the chromosome N in the wild wheat. The short arm of this chromosome is responsible for producing these nitrification inhibitors in the wild wheat. This is transferred into the cultivated wheat, initially using the Chinese spring as one of the model species of the cultivated genetic stocks. And then eventually it is transferred into the elite wheat varieties of this is a monal, high yielding elite wheat variety that has a yield potential of about 10 tons per hectare grain yield. And this is the wheat limus translocation chromosome that is introduced into the limus, into the monal wheat variety. This is before introducing this wheat variety. This is how the karyogram looks. This is after introducing this translocation chromosome. This is how the BNI elite wheat variety looks. Because of this, the wheat variety, the new wheat variety we call BNI wheats, can produce large amounts of antibiotics from the root systems. You can see the Before introduction, this is the amount of antibiotics the nitrification inhibitors produced from the wheat root systems. This is after introducing the translocation chromosome controlling the inhibitor production, nearly 2 to 5 fold increase in terms of the nitrification inhibitor production from the root systems we can see. So, because of that, because of this inhibitor production increase with these BNI wheats, the biological nitrification inhibitor capacity are enabled wheats. They produce less nitrates, and also because of the controlling the nitrification and the less nitrate production, they also emit less nitrous oxide emissions. We can see because of that, the BNI wheats produce less nitrate compared to the current elite wheats varieties, about 30% less nitrate production we can see. And you can see they emit less nitrous oxide emissions from the wheat fields because of this enhanced nitrification inhibitor production from the root systems. And this is the net result what you see in the field. What you see on your left side is the BNI monal, the elite wheat variety where we have introduced the BNI trait from the wild wheat, that is the chromosome segment controlling the inhibitor production. And on your right side, this is the same wheat variety, the monal, before introduction of the BNI trait. So with the BNI trait, without BNI trait. So with the BNI trait, because of this enhanced inhibitor production, the nitrate activity is controlled, and the amount of nitrate production is less in this BNI wheats, and the amount of nitrous oxide emission is less because of the BNI trait, and that's the reason nitrogen remains in the soil in the BNI wheats, whereas in the non-BNI wheats, that is the currently available, most of the modern wheats are the non-BNI wheats, the nitrogen cannot be retained. That's the reason you can see the nitrogen deficiency symptoms here. And that's the reason you need to apply more and more nitrogen to keep them green and productive. And if you see the grain yields of the BNI wheats, this is the BNI wheat. This is this is the BNI wheat. This is the non-BNI wheat. The same. These are used the isogenic lines before introduction of the BNI trait, after introduction of the BNI trait. We can see the BNI wheats consistently produce higher yield compared to the current available, the current variety of the elite variety of wheat. So the BNI wheats can produce almost similar yields of grain yield of almost nearly 50% reduced nitrogen fertilizer compared to non-BNI wheats, the current modern wheats. So basically what these BNI wheats are doing is they are producing large amounts of these antibiotic compounds that are specifically directed at these nitrifying bacteria that are involved in using of this nitrogen fertilizer and then converting them into nitrate and nitrous oxide. So this kind of bacteria are controlled in the BNI wheat rise in the root zone. That's the reason the nitrogen remains in the root zone and is available for the plant, for the crop uptake. So this is called the BNI wheat technology, what we are now developing. Because this is a seed-based technology. Once you develop these new category of wheats, these BNI wheats, then this technology can be easily scaled up. This can be transferred. This translocation chromosome, what you see here, this is the translocation chromosome, which is a part of this chromosome coming from the wild wheat. This translocation chromosome can be transferred to any of the other elite wheat varieties that are grown in different parts of the world, and so that the technology can be easily adopted and scaled without much effort and additional cost to the farmers. And by introducing the BNI trait, you can also see the nitrogen uptake is much more enhanced in the BNI wheats at various nitrogen applications compared to the normal wheats. And also, as you can see, the yield levels also have improved because at various nitrogen applications, because the BNA trait, when we introduce into the wheat, it produces the nitrification inhibitors from the root systems, but also it facilitates extraction of organic nitrogen from the soil more efficiently than the normal, the current wheat varieties. Because of that, even under very low nitrogen applications, the BNA wheats extracted more nitrogen compared to the current light wheat varieties. Because of that, the BNA wheats can perform reasonably better performance at various nitrogen applications. So, and also they can yield probably as good as the current wheat varieties with 30 to 50% less nitrogen fertilizer applications, because the BNA wheats, because of their ability to produce this kind of nitrification inhibitors in large amounts, the nitrogen leakage from the wheat fields is substantially reduced. Because of that, you don't need to apply so much nitrogen fertilizer. That's the reason they can perform equally. They can perform as well as similar to the light wheat varieties with 30 to 50% less nitrogen applications. And this technology now we are trying to introduce into the wheat varieties of Indo-Gangetic plains, particularly focusing on India. So this is the new satraps project started this April. Yeah, so recently, this year, just a few months back, this was started. This is the Indo-Gangetic plains. What you see here, this is the major wheat growing region in India. So we are trying to introduce this BNI trait, basically by transferring this translocation chromosome into the elite Indian wheat varieties. These are the Indian wheat varieties that are selected to transfer this ability into these Indian wheat varieties, so that this can have a very big impact on in reducing the nitrogen fertilizer usage in Indo-Gangetic plains, which is the main the wheat growing region in India. Since as I mentioned, this new technology can be easily scalable and easy for the farmers to adopt. So this technology can be probably once we develop once we demonstrate that this technology works in a large scale forms, then this can be easily adopted by the farmers, not only in India, but across the world. We have several collaborators from Europe and from US and from other parts of the world working with us in transferring this BNI translocation into the elite wheat varieties that are grown in different parts of the world, because this technology can be easily scaled up and it can easily be adopted in different wheat growing regions of the world. And also we are trying to introduce this new technology into the Nepal, where some of the elite wheat varieties that are already carrying this BNI trait like Munal are well adapted to the Nepal conditions. So now we have started recently to test this whether the BNI wheat technology works for the Nepal conditions. And so this is probably one of the new technologies that can have a very big impact in terms of reducing the nitrogen fertilizer leakage from the wheat production systems. Thank you for your attention.